Hello, everyone. I'm very excited to be able to share with you my research on the phylogenetics and classification of the fireflies. And I'm extremely humbled and grateful to the awards committee for selecting this research for the Lacordaire Prize. Now, I'd like to start off with this quote by Barber. Taxonomy from old mummies, which fill collections, is a misguided concept. It leads to the misidentification of rotten samples in collections. How these poor fireflies would resent being placed in such diverse company among specimens of enemy species if they were alive and intelligent. What contempt they would feel for the damned taxonomist. Now, in terms of fireflies, one of the things that I really love about them is their diversity. So we all know about their ability to bioluminesce, but one thing that's really interesting is the various ways, both behaviorally and morphologically, that they achieve this bioluminescence. Another interesting aspect of their biology is that not all fireflies rely solely on the use of bioluminescent communication. Many of them still rely on pheromone usage, and this is reflected in the various morphologies in their antennae. And so, one interesting thing that this leads us to is disagreement in their classification. Depending on which set of morphological characters we use, we can either get similar classifications, such as these first four subfamilies, or we can get vastly different classifications. And sometimes we can't even agree on what a firefly actually is or is not. And so the objectives of this research were to use an anchored hybrid enrichment approach to infer a phylogenetic backbone. We wanted to use that backbone to identify any areas of non-monophyly specifically at the subfamily level and update that classification. So in order to do this, for our outgroup, we chose to route to an omethid, and we included all of the hypothesized sister taxa to the Lampyridae, including lycids, cantharids, elaterids, and phingodids. So for our in-group sampling, we had about 88 species representing 53 genera and 7 out of the 8 subfamilies according to Crowson. Now, the probes that we used for our anchored hybrid enrichment were designed by Katrine Stenger Hall in her lab at the University of Georgia. So they started with a subset of 941 Coleoptera loci. They added to that data set 17 Lampyrid genomes and transcriptomes and got a resulting 516 target loci across the Elateroidea. So the anchored hybrid enrichment data was generated by the Lemon Lab at Florida State. And given their benchmarks and metrics, specifically getting orthologous sets with more than 50% species representation, we were left with 436 loci across our Elateroidea sampling. Now, once these loci were aligned and mapped, that gave us quite a large alignment of 1.8 million base pairs. And we really wanted to look at the data a little bit more in depth and see what we could do to the data and see how our data held up in terms of our phylogenetic inference. So we took the pipeline of Breinholt et al. 2017, and we trimmed the columns of our alignment based on their density and their entropy. So how random the data appeared. And what this left us with was an alignment of 401,000 base pairs. So quite a bit of difference between our two data sets. And then we ran both of these data sets through two different phylogenetic inference models. The first of those being maximum likelihood, where we used IQ tree. The second being more of a coalescent model where we utilized astral. And for this astral analysis, we did infer the 436 individual gene trees via IQ tree first. And if anybody has any extra questions about these methods, I'd be more than happy to discuss that later, during, or after our meeting. The important part of these various methods is that there was high congruence between the subfamilial placements. And so for this research, Given our taxon sampling, we really weren't looking any more deeply than the subfamily. And so we had high confidence in our decisions regarding the classification. So since there is this congruence, for the rest of the talk, I'll just be referencing and showing the maximum likelihood tree, which was based on uh, the full 1.8 million base pair data set. So when we zoom in here on the phylogeny, the first thing to note is the sister group relationship with the fireflies, which in our hypothesis would be a mix of Elateridae, Phengodidae, and Rhagophthalmidae. Now, within the fireflies, the first thing that I want to point out here is this red clade with the dotted black line. So this red clade represents the Luciolinae, which so far in firefly classification has been the most stable subfamily. And this subfamily is recovered in all phylogenetic estimates, and it's defined based on the head not being covered by the pronotum and the males having 6 visible abdominal ventrites. So what about that dashed black line? Well, that dashed black line is the genus Lampyridae. The reason that it's dashed and not solid red is because given the large difference in morphology based with the typical Luciolinae, we were uncomfortable making a taxonomic change without looking at a broader sampling of the Lucioline genera. Um, so just to note, the Lampyridae, their head is covered by the pronotum. Their males have 7 visible abdominal ventrites, which is why they have typically been classified with the Lampyridae subfamily. However, it seems based on these 436 loci that they should reside within the Luciolinae. If we continue on in our phylogeny, we get to a couple of really interesting groups. So in yellow, we have what we're calling the Lampyridae. In pink, we have the Psilocladiinae. And in orange, we have our limited Amiditinae. Okay, and so if we start with that yellow clade and really focus in, we can see there are 2 different genera here. And they're not just the ones with light organs and the ones without light organs. Okay, so we have here the Phausis, which are typically an American genus, a North American genus, and the Lampyridae, which are typically a European genus. Now, over the past few decades, these 2 genera, based on similar morphology, have been lumped and split into in various combinations. Now, based on our phylogeny and our estimate, it looks like the 2 of them are closely related. And so we're suggesting this new subfamily, the Lampyridae, defined based on the mandibles not being reduced in size, based on the antennae being filiform with or without a terminal sensorum, with simple tarsal claws, 7 to 8 abdominal ventrites, dorsal abdominal spiracles, and with a symmetrical ediagal sheath. Now, if we move on to that pink clade, we get to a very interesting clade, one in which I'm hoping to continue researching because I feel like we don't have all of the answers yet. And so previously, this clade was the Siphonocerinae, which included Psilocladus, Poloclassus, and the Siphonoceras. However, in our analysis, we were unable to sample Siphonoceras, and neither Poloclassus was recovered sister to the Psilocladus. For that reason, we're choosing to re-erect the Psilocladiinae subfamily, but restrict it just to the Psilocladus genus, and define that based on the antennae having 11 articles, with articles 2 through 11 having weak ciliate branches. Okay. Further in, we have a now monophyletic Lampyridae based on several Amiditine genera being recovered within this clade. And what this shows us is that the reliance on the antennal features is really misguiding our evolutionary analyses. And looking at Lampyrid systematics in a more broad scale, utilizing more information, is going to lead to a better understanding of the evolution of this group. Okay. And so, in summary, our taxonomic changes were that we re-erected the Psiloclidini, we erected the Lampyridae. We limited the Amiditini to just the Amidates, Magnoculus, and Momoan, and primarily transferred the Cicicauda to Lampyrini. Unfortunately, several genera we were either unable to sample or they had questionable positions within the topology or with regard to related genera. And so we elected to leave these genera in Certocetus and look at them in future research. And so before this research, we had three monophyletic subfamilies, the Odotrotini, the Teratini, and the Chegevarini. After this research, not only were those three subfamilies recovered monophyletic, but we were able to add the Siphonocerini, Lampyrizini, Psiloclidini, Amiditini, and the Lampyrini. So what this research shows is that these probes, these anchored hybrid enrichment probes, are efficient across the Elateroideum. And these probes are available on our data dryad links. And we are very hopeful that more researchers will use these probes and link them to this data set to give us an even broader taxon sampling and a better understanding of the evolution of fireflies. Okay. Additionally, it's not enough just to use morphology or just to use phylogenomics. It's once those two come together and we're utilizing more information that we get an increased understanding. And hopefully in the future, we can also incorporate behavioral differences and more ecological traits to help further our understanding. Okay. We also need further attention, specifically within the Luciolini regarding the genus Lamprigera and the Photorhini regarding the genus Vesta. With that, I'd like to acknowledge all of the labs, people, curators, collection managers that really helped promote this research and really helped us get a better understanding of what's happening within the Lampyridae. And with that, I think I have time for questions.